So, welcome back to this course on functional genomics. In the previous lecture, we looked into the various databases that are used to store the information that are derived from the genome, including the functional aspect and how you can query them to understand the function of genes and build your hypothesis, do more studies to understand the function of the genes. So, in this class, we are going to look into some of the methods that have been developed in the early years to understand or to sequence the genome. So, let us ask the question like how the genome is so special for human such that we became a species which study the other species. So, for example, dog do not go to the university or college and study science and use human as a model system, but we do it other way. So, what is so special about the human genome that made us so successful species in terms of your cognitive abilities? You are so successful, you could domesticate other species and study them as a model system or for our own benefit. So, what is shown here in this slide is the range of sizes of the genome of representative groups in log scale. So, that is what is shown here. So, you can see here that human is here. The genome size is comparable amongst all the mammals because we believe that the humans evolved from a common set of species. And if you compare the genome size of the humans with the other popular models that we study, it is not that the genome size correlates with the complexity. So, you can have, for example, a frog which you call a salamander, which is having a genome which is several fold larger than human. Likewise, you have plants, for example, the lily, the genome size is much larger as compared to the humans. So, ours is not so different from the chicken or the toad or even, for example, the fly and so on. So, what is that makes us so special, right, that become so successful species and we study the other species? This has been the query for the scientists for a very long time, and they try to investigate. One of the earliest theory was that we have our genome size is larger, that is why they looked at the genome size, and this can be easily done by measuring the amount of DNA without really knowing what combination of bases and sequence and so on. So, that is what is shown in the previous slide. Then people thought it could be the number of genes, you could have more genes as compared to other species, that is what is shown here. So, with the sequence of the genome, we sort of predicted the number of genes that we have. The human and, for example, the mouse, we roughly calculate about 20 to 25,000 genes that we have. It is not very different significantly as compared to, for example, nematode. It has got about 19,000 genes, and fruit fly is about 13,500 genes. So, that is it looks like it is not really the gene number per se that makes the huge difference, and of course, we have the genome size is much larger as compared to the other species. Then with other approaches that we will be talking about a little later is by sequencing the transcripts. Now, we understand that possibly the difference is in the transcript. So, you can see here the total, if you look into the relative proportion of the genome that is transcribed into RNA that do not have any coding potential, meaning they may not really code for genes, so they are not classified as genes, that really is gone up. For example, it is 33% of the genome, but as you come up to the human, it is much higher. And if you look into the ratio of non-coding sequence that is shown here is gone up here, all right. So, it really shows that from a nematode or fly, which is if you look into that coding versus non-coding, which is not very different for these two groups, but as you come to highly evolved species like mouse and human, you find that complexity has gone up. So, probably the difference is the genome size, of course, plus the proportion of the genomes that code for transcripts that do not code for any protein. So, these are what is called as non-coding transcripts, which have very critical function, and some of which we have already discussed in the previous lectures. But for majority we really do not know what is the function, that is something being investigated. So what is your genome? So if you look into, it is very different from for example microbe. You have in the bacteria you have the circular genome, but in humans you have distinct chromosomes like what is shown here, 23 pairs, and if you look into the chromosomes, then the genes are spread out all over the chromosomes, and that is something that is shown here. So let us see how the genome sequence began and what are the techniques people have used and how it has changed over time. So in 1972, the first sequence, that is 24 base pair, was published. A small effort, and they came up with an approach to sequence part of sequence, and then the Sanger sequencing was invented. When that has really changed the way that the sequence, DNA sequence was done. And one of the prerequisite for the Sanger sequencing is you have to start with DNA that are identical, multiple copies of a given segment of the DNA, then only your sequencing would be good. So we will see why it is so. And that was a limitation, because then you have to clone every small segment and make multiple copies of it and then sequence. But with the invention of PCR approach, it has changed the way the Sanger sequencing is done, because now you can amplify, meaning make millions of copies of any small region of the genome, and this PCR product can straight away be sequenced. So that came what is called a cycle sequencing, like using PCR approach to sequence the genome. So that has led to what is called as automated DNA sequencers, where you set up the reaction and leave it, and the sequencer machine would run and decipher the sequence and give you as a data. This again was something undergone one more round of evolution and revolution. By 1995, instead of classic gel based sequencers we will discuss, we have what you call as capillary. So you can do 96 sequence runs at the same time, but everything has now is changed with the introduction of what is called as next generation sequencing, or sometimes this people call as second generation, because the Sanger based Sanger method is called first generation, and we have now second generation, which you call as next generation. But now it's also a new approach coming up, which is called as the third generation. But as of now, this is called as the NGS or next generation sequencing. But it's not that the DNA sequence and sequence approach that came first. In fact, people started using RNA much before the DNA could be sequenced. Most often the choice was reversible RNA or virus RNA, because then you have the RNA in multiple copies. These are identical sequences, you have multiple copies you could sequence, but that was the challenge with the DNA. So how would you isolate a small segment and make multiple copies? That approach was possible because of the cloning techniques that evolved, that we discussed, called as recombinant DNA technology. And because of the RNA sequence, chemical based sequencing was possible, that was developed by Walter in the early 73 or so. This group is able to publish one of the bacteriophage, viral bacteriophage, which has got RNA genome. They are able to sequence and publish it in 1976. So that was the first major sequence initiative, and by then the other people, including Sanger, invented this approach. That's what you're going to discuss. So, but after the viral RNA sequence was completed, and then Maxam and Gilbert, they together used very similar approach to sequence the DNA. This is called as chemical sequencing, and they use a very similar approach. But by then, you have the recombinant DNA methods have come in, and therefore you are able to have a copy of the small segment of DNA, multiple copies you could have, so you can sequence it. But as I told you, that the automated DNA sequencer are possible because now we are able to fluorescently label the 4 bases of the DNA. All these that we are talking about from here to here, these approaches use radioactive moiety attached to one of the 4 bases or all the 4 bases, and that is where you are able to see the sequence of the DNA or RNA, because you are labeling the DNA or the bases. And then by 1985, we have had inventions in which they developed what is called as fluorescent moieties that are attached to the bases, and that was pretty successful with the Sanger method. We will see little later, and that has changed the way that sequence is done. That is what led to what is called as automated DNA sequencing. So this is the first paper that we talked about, the Maxam-Gilbert sequencing, a paper published in 1977, a new method for sequencing DNA. So they use the chemical method that is shown here. So you basically have a short stretch of DNA, and the DNA is put into, for example, 4 tubes, and each in tube you are adding different chemicals. These are methods that would cut the DNA at different bases. For example, this method, what is shown here, would cleave the DNA, and that has in a point where you have a G or A, whereas this chemical method would cleave the DNA at A, and this one wherever C and T, and this one wherever you have T. As a result, you could have sequences. So basically what is happening is that you have the DNA that are labeled at one end with radioactive moieties. For example, you have a DNA, and this DNA can be radioactively labeled by a method. For example, this is your DNA, and you have 5 prime, you have 3 prime. So you can add a phosphate group here, which is attached to for example, radioactive moiety by using a kinase, and this would do it on either end, but after that you can cut with a restriction enzyme and take only this part. So in this fragment, only one of the 2 strands is labeled with the radioactive moiety, and then you use this chemical method. Therefore, this would cleave either here, 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 here based on where it happens. It is a random event, and therefore, when you run a gel, you are going to separate the DNA fragment based on the size, and what you are looking at is only the radioactively labeled DNA using X-ray film. So that is what is shown here. So you, by looking at the sequence, you are able to read the, looking at these bands, you will be able to read the sequence. For example, I have a band here and here that shows that what I am seeing here is C, because it is present in the C lane as well, but the one that is here present only in this lane, those cannot be C, therefore you call it as T, again C, and then likewise you go on reading the sequence. So this is the first method for developing or understanding the sequence of the DNA. But still it was very cumbersome. So this gentleman, what you are shown on the right side, is Sanger, who developed a method for sequencing DNA, which is based on very similar principle that our cells use to copy the DNA. So as you must have studied, for the DNA to replicate, you have an enzyme called DNA polymerase, which reads one of the single strands and keep adding the complementary bases to make a new DNA, and that is the method that he pretty much used to sequence. So what is unique in that? So he sort of chemically modified the base such that that base, if it gets added, the polymerase will not be able to continue the polymerization. In other words, you terminate the polymerization. So these bases are added in a smaller amount, therefore you have a normal base with small amount of this modified base which terminate, and as a result you would not terminate the polymerization at every point, but in certain points. That is what the method was, and in fact he was awarded Nobel Prize for this particular invention, because it has really changed the way the DNA has been sequenced. So first paper that came in 1977, this is a nucleated sequence of bacteriophage DNA, right. So this is called as Sanger's dideoxy method or Sanger method or chain termination chemistry. There are several methods. Normally it is called as Sanger method. So this is a normal base. So you have the sugar and then the nitrogen base. So what he developed is an analog of this base, which is called as dideoxy, meaning you can see here you have for the DNA in this sugar you have OH as well as here, but here it has been removed, right. So, that is what it is. So, if you look into the way the DNA is being made, you can see here that every base is connected the way it is shown here, right. You have the bonding. Whereas, if you have a base which has got dideoxy sugar, then this if there is a polymerization, there is a DNA polymerase that help in copying the strand that is shown on the right side, then this base would terminate the polymerization because it cannot really help in continuing the polymer because it does not have an oxygen over there. So, that is the invention, and that really changed the way it is done. So, it is using the same the same machinery that our cells uses, because you have the template DNA. So, what you give is a small primer, an oligonucleotide that is complementary to the known sequence. It goes and binds, and then you have this DNA polymerase comes and sits here, and when you have all the 4 bases available, it is going to just keep on copying it until it incorporates a base that is shown here, right. So, if that comes here, then it is going to terminate. Let us see how does it happen. So, there are several methods by which you are able to approaches by which you are able to read the new DNA strand that is being made. One of them is that you label the primer itself. So, what is shown here. So, you have the 5' end of the primers labeled with, for example, radioactive moiety. Therefore, every new fragment that is being made, you are able to detect in a gel, right. Or you could use in the reaction bases that are labeled. For example, the 4 bases that you are using, you can use them in a labeled way, so that every base that gets incorporated has got a radioactive moiety. So, therefore, it would give you the signal when you are visualizing. The other method that has developed is that whatever the modified base that are using for termination, the dideoxy 1, that alone is labeled. So, as and when it the DNA gets, the growing strand gets terminated, that would have the dideoxy 1 as well as it will have the radioactive moiety. Therefore, you are able to visualize them on in the gel. So, these are the ways that you do it, and this is what the reaction is. So, you have the primer which is labeled, let us say that is what we are talking about, this particular approach, and then the primer goes and binds to, and then you have the DNA polymerase which copies it, and depending on when do you get this dideoxy base, it would be arrested. So, when you separate the fragments on a gel with separated, then you could really read the sequence. So, this is the smallest fragment and this is the largest fragment, and you can see that this lane represent a reaction in which you have used a dideoxy T or C or G or A. So, by looking at the fragment, you will be able to read it. You read it as, for example, this is T, G, C, G, 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 C, and so on. So, that is what it is shown here, and this is the sequence of the DNA that is being made, and this is the sequence, the reverse complement is the sequence of the original DNA that was used as a template to sequence, right. So, in this way we are able to sequence the DNA, and that is a remarkable invention, and the way we have done is evolved over time. This is some of the classic manual sequencers for radioactive gels. So, you have to apply the samples here, and as I told you, there are 4 lanes for each DNA, A, G, T, C, and separate them like what is shown here, and this is one such example of the gel sequence after autoradiography. After separating the DNA, put an X-ray film wherever you have a radioactive moiety, you would have this kind of black color. Now, you are able to read it based on this, right. So, this is how it is done. But things have changed, as I told you, in 1995, 94, different chemistry have come in. One of them is to use what is called as a non-radioactive method. Instead of using radioactive moieties, now people started using fluorochromes, different chemicals that can, when you excite with a laser, then they fluoresce in different wavelength. Therefore, you are able to distinguish one base from the other. So, in other words, you can do all 4 reactions A, T, G, C, termination in any one of the bases in single tube, and depending on what kind of fluorescence they emit upon excitation, you will be able to call it as which base it is. So, in other words, what you have done is, instead of loading 4 independent lanes for a given DNA, so now you have a gel in which each lane represent one sample, and you are separating it as the DNA pass through at the end of the gel. So, you have a laser which excites, and then you have a camera which basically measures the fluorescent intensity at different wavelengths. For example, you have a filter which basically looks at the emission at different wavelength, and depending on in which filter you are able to get the signal, you will be able to call it as A, T, G or C. So, in this way, it is taken care because you load the gel and forget about it, the rest the machine will take care. And so, you have a camera that reads here, in one lane you can do all the 4 reactions, and at the end of the run you have this kind of electrophotogram which talks about the intensity, the peak intensity for a given fluorochrome in a given segment, and this is one base and this is another base. If both are giving a maximum fluorescence that denotes C, then the machine calls it as C, and if likewise you have these sequences, right. So, this is how you are able to read the sequence. It has changed. So, this is the first generation automated DNA sequencers, right. So, still you use gel, cast gel, and you have to load sample, do a reaction and load sample, but separation and reading the sequence was automated. So, it really changed, but things have gone much better. So, this is the method that we just now discussed, that you have a slab gel which you have to cast and run the sample, but then people have gone to use a different method called as capillary. Instead of gels, you have thin wires now which have hole inside, in which you can fill the gel and allow the system to pass through, do the electrophoresis of the DNA, and when they are coming out, you read what is the fluorescence that comes, and you are able to read it. So, this is called as capillary DNA sequencer, something like that what is shown here. So, you have done away with the gel. So, you no longer you have to make the gel, and you have thin capillaries. Each one now can go and take the samples, separate them and read out. So, you can increase the number of capillaries, you can increase the number of samples that you would analyze. Not only that, once the sample is read through, then you can remove the gel and fill in new set of gel, and you can start doing another reaction. So, in this way you can automate it better as compared to this gel which normal traditional way of slab gels. So, that has really increased the throughput, like number of samples that one can analyze and so on. So, that has changed, and in fact that has led to faster ways of sequencing the human genome. So, the capillary sequence has really helped in hastening the speed at which the sequence of the human genome was done. This is one such example. So, that is a DNA capillary electrophoresis, which again you have all the sample that been kept here, and then you have capillary that is running here for the sequence to do. So, that is what we discussed. So, you have the fluorescently labeled dideoxy NTPs change the way for automated sequencer, and then you have the capillary sequencers have come, and so it has changed. But it is not only the chemistry of sequencing that has changed, even how do you prepare the DNA and how do you analyze the sequence over time to get the sequence assembly, that has changed. So, this is one is Human Genome Project that has used a different way of getting small segments of DNA for sequencing and how to analyze it. And then in the same time, we talked about the other person called Craig Venter, who developed a different method of sequencing, which is called a shotgun sequencing. What he has done is he has used the influenza genome to sequence using the shotgun sequencing method, which we will discuss about, and he has then used his the same method to look at human genome sequencing. So, the way it has been done has dramatically changed over the course of time. So, as a result, you can see that if you look into the percent of the human genome sequenced, you can see that when the automated sequencers came in and when the capillary sequencers came in 1995, somewhere around that, the rate at which the genome sequence was completed has dramatically changed. So that really helps us to understand how the technology has transformed the way we look at the sequence. Let us look into the two different approaches people have used. The Human Genome Consortium used an approach called hierarchical shotgun sequencing. So basically what they do is that, or what they have done is that they have isolated the genomic DNA, and then the DNA is digested with restriction enzyme, and then the fragments are cloned into vectors called as BAC, which stands for bacterial artificial chromosome. So they created a library, meaning the bacterium has got different segments of the human genome and different cell, right? And after that you take one of the BACs and then screen for the library, and you are able to identify the BAC clones that have overlapping DNA segments. So they are able to get different pieces like puzzle piece that overlap with each other. They are able to first make what is called as contic, right? These has got overlapping regions, and then each clone was digested, cut into smaller pieces, and then they were sequenced. And what you have done is we have each segment that was sequenced. Now you try to align them, get a larger sequence, and then go and align with the other, and that's how they are able to assemble this. Okay, so this is a method that the Human Genome Project Consortium used, and it was time consuming because you have to create a library. But the Celera, the private organization that sequenced the human genome, used a different model. This is called a shotgun sequencing approach, wherein they basically fragmented the DNA into smaller pieces. They went on reading each base the sequence. So, and then based on the sequence they used a powerful computing tool to identify the sequence that overlap, and they get the consensus. And then you try to use different methods to sort of identify sequence that could match each other and make a much larger sequence. What they have done is there are several gaps in their approach, but they have used the reference sequence that were given by the Human Genome Consortium. They used it and quickly they filled in the gaps, and they are able to complete the sequence based on the publicly available data. So that's the other approach that they have used. So based on that, you have all these sequence now. So now you can look into where is the transcribed region, where is the region that codes for the protein, where is the region that are regulatory. The different elements that we have seen in the previous lecture all are derived from this kind of approach, right? So that is how led to this kind of genome browsers, right, which gives you all the information. Expectedly, with we can see that from 80s to 95, there is a dramatic increase in the sequence and that are deposited in the gene bank, and of course with in the last one decade it has really changed. What is shown here is the log scale. You have to understand it is going to exponentially is increased what being deposited, right? So that really helps us. So what is the outcome of the Human Genome Project? Is it simply a kind of a mission that completed showing that we can do something, or it really made any difference to us? So it has hastened the sequencing of other organisms. Now we are able to, we have, this is somewhat outdated, we have 40 plus model genome sequenced, more than 2000 microbial genome sequenced, 2000 viral genome sequenced, and a large initiative we discussed in the previous lecture, what is called the metagenomics initiative, where you try to understand the genome of all the microbes that are around us, including our digestive system. So this helps us to understand the relations, the conservation and the gene functions and so on, and enabled multiple genome related projects. For example, the ENCODE, AppMap, for example, human genome variation projects, the genome-wide association studies to identify genetic risk factors and understand the genome diversity across human population. All these things have happened because of the technologies developed, because of the understanding that we have on the genome. And then obviously one of the important outcome is that the databases, we have now gene ontology. If you have identified a gene, then you are quickly able to understand the function that we derive from other model systems. We have gene bank, genome browsers and so on. And of course, the new generation of techniques, right? That is what one of the greatest outcome, development of what is called as next generation sequencing, RNA sequencing, ChIP-seq and many others that we are going to discuss now.